in Georgia. I'm at the Joint Department of Biomedical Engineering, joint between Georgia Tech and Emory University. So my chair reports to both deans, the Dean of School of Medicine and the Dean of Engineering. So really allows us to kind of explore that space. But I have been running the Platt Lab for repair, regeneration, and remodeling. Again, cardiovascular tissue remodeling is a major research area of mine. Again, today I'll be talking about sickle cell disease, and particularly our interests are why children with sickle cell disease are at increased risk of strokes. I did a lot of work in the past with HIV and cardiovascular disease. Funding kind of has dried up, but we still keep one leg in the idea space there. And I do some work with system biology and predictive medicine, so they keep me in an engineering department. Okay, so, but check us out, plattlab.com. Looking forward to sharing this. But when I say we do tissue remodeling, I like to say what that means to me. So that look at how do you go from this healthy tissue to this disease, atherosclerotic tissue. So this is again for this audience especially, nose is a human coronary artery, and you can see blood will be coming out of the board. And what are these processes that lead to this atherosclerotic plaque that is developed? Well, some of those initial stages are protein degradation of some of the structural components of the artery wall that then the cells then respond to the breakdown of that structure, and cells can infiltrate into this growing neointimal space, synthesize new proteins, deposit new proteins, positive feedback, etc. Another example again is osteoporosis, where this normal bone matrix is highly collagenous and mineralized for both biomechanics, but also we know it serves as a calcium reservoir. But then for osteoporosis happens more commonly in postmenopausal women. There's loss of that type 1 collagen component, demineralization. And again, not only do you lose the biomechanical components of the bone, but also some of the biochemical components. This was one of the walking biobots from this NSF center I'm a part of called Emergent Behaviors of Integrated Cellular Systems. It was a skeletal muscle bot on these PDMS feet that when you applied electric voltage, the muscle would contract and it would walk across the screen. And this is developed by Rashid Bashir's lab at University of Illinois. My job on the project was looking at over time, the bot would stop walking and break in the middle. And so we wanted to look at what are these processes that causing it to remodel this tissue and space to break, which again, there's tension from the force in the walking, and then there's just biochemistry of expression of proteases that break it down. So again, how do you just change tissue from one structure to the next? And that's what takes me to sickle cell disease. My PhD work was all about atherosclerosis by shear stress, endothelial cells, proteases, the usual things. I call them usual, but no, they're really exciting, very cool things. But then when I started my independent group, I was exposed to people working on sickle cell disease, the disease we've all heard about, biology class in high school, and it's one you can talk about. But it's not cured. And some people do think there's a cure for sickle cell, and it is not. It's made its way back in the public lexicon with genome editing technologies recent times. But I like to call it the singularity of sickle cell disease, which again, it's a single point mutation. And, back in the day, we learned that a point mutation can be a problem, but major gene problems were frame shifts and all the other stuff. But a single point mutation changed an A to a T. Of course, that will then change one amino acid, a glutamic acid to a valine, which again, it's in one protein, hemoglobin. But here's the thing about hemoglobin. Hemoglobin is only in one type of cell.
My licensed doctor had SDS, so I added potassium acetate because that's
okay. So hemoglobin is only in one type of cell, red blood cell. A lot of the other protein mutations we study, they're expressed in multiple cell types, so it makes it even more complex. One cell. But what's the problem? Red blood cells, they go everywhere, right? And so that's why you get this multifactorial disease. And in the United States, it still is a health disparity, but it also still causes reduced life. So life expectancy for men is 36 and 42 for women. But it's a health disparity in the US. 1 in 400 African Americans have sickle cell disease, and 1 in 12 have sickle trait, meaning they're heterozygous. So sickle cell is a homozygous recessive disease, get the 2 mutant alleles. And the 1 in 12 have, again, the 25% chance of passing it on to their offspring if they reproduce with someone else that's a carrier. So it's also the most commonly inherited blood genetic disorder in the United States. There are 100,000 Americans with sickle cell disease at common estimates. But there are millions more worldwide. This is a global health problem. So there are 300,000 babies born every year with sickle cell disease. And this exploded cartogram kind of shows that in sub-Saharan Africa is a major population, the Indian subcontinent as well, huge population, the Arabic world, and then you can see North and South America, but this is believed to be because of the international slave trade of the 1700s. But the real sad part to me is that up to 90% of these children will die in the first 5 years of their life. usually due to infection. So if you give children penicillin from birth through the age of 6, then they can survive. But again, in low resource settings where that's maybe not diagnosed at birth or diagnosed before, then these complications arise. So this is a global figure, not the US figure. But that's why also when I talk about sickle cell disease, I talk about the importance of being a scientist activist and telling people what's going on, particularly as this affects Black Americans more so in the United States who may not know their status, right, or know if they're a carrier. Everyone is tested at birth, but they are told maybe that the sickle trait to be heterozygous is a benign condition and you don't need to worry about it, but that's not officially true. So again, I also think globally, act locally. This was a slogan I used to see on my after school programs in the 80s and early 90s. But again, the world shrunk for a while. So I say then act globally. Now the world has expanded again with COVID-19 and quarantine, but we'll get back there. But I think it's again important to remember that global health concerns are health disparities for underrepresented minorities in the United States. And I really started to learn this when I was in the HIV/AIDS research world, where they wouldn't want to talk about in the US, but some of the HIV/AIDS rates that they were focusing on in developing countries were the same rates for African Americans here at home. And again, understand this importance of being a scientist activist when studying health disparities to make sure you can communicate with the different groups that these things might be impacting. So back to the sickle cell and the molecular origins. So that mutation, that single point mutation in hemoglobin, it causes these red cells to take on a sickled shape. Why? That glutamic acid was hydrophilic, was mutated out for valine, which we know is hydrophobic. So because of the shape change or conformational change that hemoglobin undergoes when it binds oxygen versus carbon dioxide, when bound to carbon dioxide, the valine is exposed on the cell on the molecule, the protein surface of the hemoglobin molecule. Remember, hemoglobin is a tetramer, 2 alpha globins, 2 beta globins, the mutations in the 2 beta globins. That valine that's exposed, thermodynamically unfavorable, hydrophobic in an aqueous environment. And so you get this rapid self-assembly in a highly ordered structure of this hemoglobin, sickle hemoglobin polymer. That also then again begins to form in these red cells when they give off oxygen and completely deforms the red blood cell. And why does it do that? Remember, the red blood cells also have ejected their nuclei. The nuclei is usually the most rigid part of the cell. They don't have one. So now it's just able to freeform inside of that cytoplasm. But as soon as that red cell gets out of, or is able to obtain oxygen again, reverse conformation, and you get melting of that sickle polymer. And this continues to happen over the life cycle of these red cells and this accumulation of membrane damage. So in a person without sickle cell, the normal red blood cells live for about 120 days. But in sickle cell, they only survive for about 9 to 10 days due to this membrane damage of stretching and relaxing until the membranes break open and lyse. That's why traditionally you may have heard about it as sickle cell anemia, because there is a reduction in the number of red cells due to the hemolysis as they circulate. But there's this beautiful images from Donna J. Call, who we lost a few years ago, showing the shape of the red blood cells. Because there's this anemia, they start to pump out immature red blood cells earlier than we normally would. So these reticulocytes, there's an increased number of these circulating reticulocytes in people with sickle cell. But he did these beautiful scanning electron micrographs in the oxygenated form or the deoxygenated form. And I show this so you can show that as the red cells are sickling and relaxing, the membrane accumulates damage, becomes more viscous, and begins to get stuck into these shapes. So the discocytes are what we normally analogize to a wild type or normal red blood cell. And you can see under deoxygenated conditions, highly sickling, and you can see highly spiculated. As it gets more sickling and accumulating damage, these dense discocytes, more viscous the membrane is, they still sickle, but you can see less shape change in the others because it's more resistant. And then ultimately you can get to these irreversibly sickled cells, where even under oxygenated conditions, when the sickle polymer is not as formed, the membrane has retained this shape because the membrane is so viscous and also quite fragile. And these are the ones that will rupture open and release those contents. But the patient indicator that's the kind of more dramatic is the sickle crisis that you may have heard about, where because of these spiculated red cells, they actually get stuck and block blood vessels. This normally happens in the post-capillary venules, because remember, in the capillaries, they give off oxygen, sickling begins, they take on these strange shapes that are not deformable, like red blood cells are highly deformable, and then they accumulate and can cause pain. And if you talk to a person with sickle, they say it's like walking around glass moving around in their blood vessels. And so they have a much higher tolerance of pain than other individuals. But when it gets to be a severe blockage, that gets extremely painful, and that's when they go to the hospital. And even though we've been studying sickle cell since 1910, currently they still just give the patient fluids and painkillers. As you all know, it's still hard to quantify pain. It's hard to localize where the pain is happening. And so these are things that, and your vascular beds are in all parts of our bodies. But there's interesting dynamics with sickle cell that make it, it's an interesting thing to study. Normal hematocrit, which this is percent red blood cells by volume, is about 45%, right? And in a person without sickle, your red blood cells will move through the capillaries, usually at the same velocity in as the velocity coming out. And the viscosity doesn't matter based on the oxygen concentration. But in a person with sickle cell, it is an anemia. They have about 25% hematocrit. So you can see a severe reduction in number of red blood cells. But what's interesting, that's a benefit to them because the whole blood rheology or whole blood viscosity is the same because they have less red blood cells. Each of these red cells is more viscous than one of these. But in that capillary, when they're giving off this oxygen, you actually get a reduction in velocity because as it's giving off oxygen, it's starting to sickle. That is slowing down its passage because it's starting to abrade the blood vessel, plus the viscosity of the cell is increasing. And so the speed coming out of the capillary is much lower than the speed it comes in. But what really got me, so there's all this work of sickle cell in the microcirculation, but when I found out that children were having strokes, it just blew my mind. Think up to 11% of children with sickle cell will have a stroke by age 20. That's a paralytic stroke, okay? With the greatest risk between the ages of 3 and 5 years old. That's crazy, right? But another third, or up to a third of children with sickle cell will have what we call a silent stroke. That's a stroke in the cognitive part of the brain. So it's not obvious as a paralytic stroke, but this Black kid may be in class getting bad grades. They're calling the kid dumb, and then they look on the MRI. Oh my goodness, they were having strokes, right? And so there's a lot of questions about why these group of patients are at risk for strokes, how to, So this is actually a magnetic resonance angiogram of a very young girl, and you can see there's an occlusion here, and this whole region of her brain is not getting oxygen. So this is a major paralytic stroke. Now, when I was studying strokes and atherosclerosis, it was like, well, that doesn't happen until the 6th, 7th, 8th decade. But this is highly accelerated happening in such young children. What are these mechanisms? Plus, the cerebral arteries are oxygenated parts of our vascular tree, not these deoxygenated parts where sickle cell is normally studied because the cells are in their sickle conformation. So there were some great images from autopsy studies from the 70s. They don't really do autopsies anymore on these young children that showed this incredible fragmentation of the elastic lamina, duplication of the elastic lamina, and then this rapid neointima that has formed here. It's been postulated there's also monocyte infiltration. It's not all just endothelial proliferation, but you see breaks in this accelerated elastic lamina in the middle cerebral artery. And here's just another image. Which I was like, wait, breaks in the elastic lamina? We know that happens in atherosclerosis. We know that cells respond to those signals and migrate. What are these parallels that could be happening in sickle cell? And so there's been some postulation again about the breaks in the elastic lamina again lead to conversion of smooth muscle cells from the quiescent to the contractile secretory phenotype. But also when we think about as that lumen is closing, normally again in a world of shear stress, as the lumen closes, shear stress increases, nitric oxide is produced, causing the smooth muscle cells to relax. Well, in sickle cell, when those red blood cells break open, they release the hemoglobin and the heme groups, and the hemoglobin actually scavenges nitric oxide. So the nitric oxide is no longer able to cause vasodilation, the smooth muscle cells. Plus, the heme group generates reactive oxygen species, which we know those are bad in the cardiovascular world. And then there's all of these things that are just put this artery wall at disfavor in sickle cell. But clinically, they've been able to identify which children are at greatest risk for stroke using transcranial Doppler. This is where the engineering comes back into play, where they can monitor middle cerebral artery centerline velocities. And if it was higher than 200 centimeters per second, these children are indicated to be put on monthly blood transfusions. That's the current state of the art if a child is at risk for stroke. Monthly blood transfusions, that's what they do, right? But that's where the engineering came in, like, well, wait, if these children are having increased centerline velocities, I had learned in my fluid mechanics class that when you have increased velocities, usually that's due to some stenosis in the vessel, whether that be due to narrowing of the lumen, due to intimal thickening, or if there's some accumulation and aggregation of sickle red cells or white cells because they're quite adhesive. And so then we started to develop these hypotheses, but also sites of disturbed flow happen post-stenosis. And we know from cardiovascular studies that disturbed flow activates endothelial cell programs that promote plaque formation and the rest. And so really this is when I first started my lab, so I really put all of these hypotheses together to understand things that I knew in the past and how we could apply them to strokes in children with sickle cell. Because again, my undergrad degree was biology. I went to get a PhD in biomedical engineering, joined a lab where they slapped this figure down in front of me and showed us that where do atherosclerosis plaques preferentially occur? At branches and bifurcations or at sharp turns. And that, the pathologist talked to the aerospace engineer buddy who said, yeah, you get these regions of flow reversal and flow separation on the outer wall, like for example, of the carotid artery. And you can see this flow circulation here. And that was then mapped to areas where the plaque would form. So fully developed flow, you don't get atherosclerotic plaques, but in regions where there's flow reversal and flow separation, plaque. But when you get back to unidirectional flow, no plaque, right? So the mechanical forces influence the gene and protein expression. And that was my PhD work. But what we identified were these class of proteases called cathepsins. These are cysteine cathepsins that were upregulated under disturbed flow and turned down under healthy unidirectional laminar shear stress. And I did this work with Hanjung Zhou at Emory. So a little bit about these proteases. If you're not studying, they are the best, okay? But they're awful to study because they're hard to study. So they were first identified in lysosomes. That's why they kind of have this moniker, because they are responsible for general protein turnover in your lysosomes. But then they started to be identified in a number of pathologies: osteoporosis, atherosclerosis. We've shown them in tendinopathy, and they had been identified in cancer and tumor metastasis. But here's why I think they're amazing. They are the most potent mammalian collagenases and elastases that we have, right? So this is critical, particularly for tissue destructive diseases where collagen and elastin are degraded. They do prefer acidic environments for their optimal pH. That's what makes them tricky. And when I was in grad school, people would say, oh, when they get outside of the cell, they don't work because it's neutral outside of the cell. We all know that now to not be true, so they don't have to stand on that case, but it's just amazing how the field moves in such a short time, because I just finished my PhD just recently. I'm quite young. No, I'm kidding. But so that's what makes them a little bit tricky. Plus, at neutral pHs, they will denature over some time frame, so it's hard to detect them. Plus, like other like digestive proteases, they are expressed as an inactive zymogen form. We call it a procathepsin that is stable at neutral pH and quite abundant, but then it has to be cleaved to be activated, and that mature form is what does the damage, all right? And Big Pharma has been aware that these cysteine cathepsins are involved in a number of diseases because they have been trying to develop and move into the medicine cabinet, small molecule inhibitors. There have been 16 small molecule inhibitors of human cathepsins that have made it into human clinical trials, and many made it to phase 2 and phase 3. And there have been 16 that have not advanced through FDA approval, which is billions of dollars lost. The most recent one was odonacotiv from Merck. Indication was for osteoporosis. And for many of these, they were effective in reducing the disease complications, but side effects are what took them out from being able to advance into the clinic. So for odonacotiv, it increased bone mineral density in postmenopausal women with osteoporosis by 68%. And this was 16,000 patients over 12 years, 8,000 in each arm, that then the side effects were, I won't talk about the side effects because it actually makes me quite upset. And I'll always spend too much time talking about them. But read the papers, I don't buy it. But anyway, okay. So these have been denied. And so we are also trying to understand what are ways these enzymes are regulated that we can use them for these diseases. So I'm gonna skip through some of the details here, but I normally have this as a nice animated slide. But I said that cysteine cathepsins are quite challenging to study because they are promiscuous. So they are denoted by letters. So K, L, S, V are the ones that I care a lot about. They are promiscuous with their substrates. They denature under neutral pH over some length of time, and the reagents and tools to study them have not been as well developed. So when I first started my lab, one of our first priorities was I wanted to make it easier for us to investigate them. So people have heard of zymography for MMPs. So we modified that protocol to get cathepsin zymography. And really what it is, it's substrate SDS page. We've impregnated our polyacrylamide gel with gelatin, just like if you're doing regular SDS page. This is a non-reducing loading buffer because we did not want to break disulfide bonds. And then after the proteins are separated, we, instead of transferring into a membrane like you would in a Western blot, we actually renature them inside of the gel by washing out the SDS and have a few other renaturing agents. So the proteins will refold in the gel at the site to which they were separated by the electrophoresis. Then we can put them into an assay buffer that's perfect for their behavior, mild, an acidic environment, mildly reducing for that cysteine at the active site. And they will cleave away that gelatin overnight at the site to which they were separated, such that we can stain with Kumasi blue the next morning. You get a dark blue background, cleared white bands indicate where your active cathepsins were. And what was really nice is the key ones that we cared about for our tissue destructive diseases, K, L, S, and V, they all migrate to different locations, such that if all of them were in one preparation, you'd be able to individually quantify these proteases. So we can do densitometry and get an amount. Why it's great for a young professor? No antibodies. That's the expensive part of Western blots, right? You just pour antibodies down the drain. No antibodies. And so we also were quickly able to find that the zymography for cathepsin K was actually more sensitive than the Western blot. This was the best antibody that was out, and I'm so sad to report it is no longer available. It's crushing. It's no longer available to any company. But zymography was able to be more sensitive. You can see the antibody petered out around 1 to 5 nanograms. Zymography way down to 0.1 and even below, which was 3.5 femtomoles of this enzyme, which was exciting. And it only detects the mature form, not the inactive pro form that's usually more abundant in systems, which can throw off your interpretation of results. Again, and we were able to multiplex it for these, and I will use it for a number of different studies in my lab. We'll talk about it here for sickle cell, but we use it for tendons, cancer, endometriosis, and anything where there is a destruction of tissue. And with a collaborator, I'm like, give us some samples. Let us run some things. But what do they do in the vascular wall? Let me speed up a bit. Again, they are able to degrade elastin and collagen in that vascular wall. They are produced by endothelial cells under shear stress, as I mentioned. Macrophages and foam cells that have infiltrated the wall also produce cysteine cathepsins, as well as the smooth muscle cells can turn on the expression of these cathepsins in disease state to then degrade the elastin themselves as they are forming the neointima. All right, so our thing with sickle cell was, all right, let's put this together. Let's look at the activity of these cysteine cathepsins in the wall of sickle cell, people with sickle cell or their arteries, and look at then what are the cellular responses responding to the biomechanics, which I mentioned is one of my areas of study, and the biochemistry, because sickle cell creates a chronic inflammatory environment because of the chronic damage to all of the tissues. So there's high levels of circulating TNF alpha and other things. So how does the biochemistry, biomechanics work together at the cellular level to then control the structure of that tissue and how it changes that then leads to some complication in the patient? And maybe we'll find some pharmaceutical targets or predictive biomarkers along the way. And so first started working with this just blood from people with sickle cell. And there's lots of studies of red blood cells with people with sickle cell. We didn't really find big changes in proteases with those. So we went to look at the peripheral blood mononuclear cells, because what is known is that people with sickle cell have increased numbers of circulating monocytes. So there's a monocytosis. And in my atherosclerosis world, we know monocytes adhere to the endothelial cells, into the wall, and mayhem continues. So we actually were able to obtain blood. I work well with the Sickle Cell Foundation of Georgia. This is Philip Keegan, the student, and Ms. Richard was the lab technician there. So we would get blood from them, isolate the peripheral blood mononuclear cells, and then we'd allow them to adhere to human aortic endothelial cells. And again, these are just repeating experiments others have done in the past. But you can see wild type is AA. That's the language for sickle cell. So this is normal wild type. When you put PBMCs from wild type individuals on ECs, you see a few stick, not much. But if we pre-stimulate the endothelial cells with TNF alpha, again, experiments we all have done, you see a greater number of adhesion of PBMCs. But with the sickle cell PBMCs, even without pre-stimulating the endothelial cells with TNF alpha, massive amounts of adhesion, right? Orders of magnitude greater. Again, others had shown this before, so we were able to replicate this in our own hands. We also tested the plasma samples to make sure there was increased TNF alpha. And you can see here, we are increased TNF alpha in the sickle cell plasma. But then the big money that got us super excited, when looking at the cathepsin activity, we can see wild type endothelials, wild type PBMCs, baseline cathepsin V and L. If we pre-stimulated the endothelial cells with TNF alpha, we see cathepsin K is now turned on, so being expressed with TNF alpha. But again, in our SS PBMCs, even without adding TNF alpha, cathepsin K is turned up higher, right? So now you're getting greater adhesion of monocytes and you're turning on cathepsin K. Oh, I didn't tell you the big thing about K. the most potent collagenase, and it's a really powerful elastase. So this is really capable of remodeling that artery wall, and it has been indicated in atherosclerosis as being turned up in plaque formation. So what are the intracellular signaling pathways that might be controlling that? So we did a bioplex on all those different conditions. And for the sake of time, I'll just feature that the J&K and its downstream target c-Jun actually showed the greatest change in those systems. And so to test for its involvement, we then went back with additional cells, some people with sickle cell, and we treated them with the J&K inhibitor SP600125 to test the hypothesis that if we inhibit J&K signaling, we'll be able to reduce cathepsin K induction after sickle PBMCs adhere. And so you can see endothelial cells alone, you don't see much change. When we add TNF alpha, cathepsin K is turned up by endothelial cells with TNF alpha. When you add J&K inhibitor, you can reduce that by about 40%. And then you see with our sickle cells alone, without the drug, you see that SSPBMCs turn up K as I showed you, but with the drug inhibitor, you can actually reduce that again by 50%, showing that this J&K pathway is playing a part in the induction of these proteases. So that's the biochemistry. Let's talk about the biomechanics. And I'll come back to the biochemistry again. But so we have these shear stress bioreactors, the Conan plates, been around for years. And we use these profiles. This is an atheroprotective one. We used to call it atheroprotective. Here we're calling it vasoprotective because we're not in atherosclerosis land without the lipids and all the rest. This is where it would be protected from plaque formation. And it would be anti-cathepsin expression here versus the pro-remodeling, which is a disturbed flow like you would see on the outer wall of the carotid artery. And this is where you would see pro-cathepsin expression in the artery wall. And so, as you all know, endothelial cells elongate and align in the direction of flow. So that works here in our vasoprotective or pro-remodeling. And these actually, we did culture these with PBMCs just for this image. But we got some really surprising results. And this is why I love biomedical engineering, because in biology, you kind of learn what happens, right? And you know, like this binds to this, this does this, and that's fantastic. But then in engineering, you have to remember cells in our bodies have to obey all the same mechanical and physical rules of like a brick falling outside, right? But then to know that our cells respond to the mechanical world, it is just fantastic. So what are we showing here is that under static conditions, these are just endothelial cells, human aortic endothelial cells, stimulate them with TNF alpha, cathepsin K is turned up, which I showed you already, right? Pro-remodeling shear stress, this is the disturbed flow that turns up proteolytic pathways. Again, without TNF alpha, we're seeing increased cathepsin K. Great, that's my dissertation work. Glad it still works. But if we add TNF alpha, of course, we get a greater amount. But this is what makes us crazy. Under the protective vasoprotective shear stress, unidirectional shear stress, even in the presence of TNF alpha that boosts that signal, you do not get the upregulation of cathepsin K in these cells. This was wild, okay? Oh, do I not show the whole deal? Oh, I did show some videos. Okay, I was like, did I show the details? So I'm going to show you what the details are happening. When I talk to other groups, I don't focus on the biochemistry much because some of them don't care. But what we found here is that even still, we went and did Western blots. J and K was still phosphorylated with TNF alpha in the static and pro-remodeling, as we expected. And so was c-Jun downstream, phosphorylated as well. Because we were thinking there was interruption of this J and K signaling pathway. So this is pro-remodeling. But when we looked at our vasoprotective indicator, even still, J and K was phosphorylated under TNF alpha. Vasoprotective shear stress even turned up J and K phosphorylation. TNF did it more. So even the vasoprotective shear stress was not even blocking downstream c-Jun phosphorylation. So what's happening here? This makes it the challenging questions to do the detective work. So then we looked at mRNA with quantitative real-time PCR, and we found that at the mRNA level, TNF alpha turns up cathepsin K. Vasoprotective shear did reduce the TNF alpha mRNA. I'm not showing it, but the pro-remodeling did not, of course. And so the question about the mRNA was, well, what's stopping the mRNA? Maybe c-Jun as a transcription factor, AP1, is not binding to the promoter. And that's why we're getting a reduction in mRNA. That was not the case. We did a chip assay for AP1 using phosphorylated c-Jun, and we were still getting binding of AP1 at the cathepsin K promoter, but we were not getting the mRNA being turned up under the shear stress. So what it ended up being, I could go through it and you can read the paper, but it ended up being that NF-kappa B is also playing a part, and NF-kappa B is regulated by vasoprotective shear stress. So under the vasoprotective shear, NF-kappa B levels were reduced in the cell. There was degradation of NF-kappa B, and you actually needed both to turn on the cathepsin K gene in these endothelial cells. And why I think that's exciting is because it toes this dynamic interplay between how the biomechanics can regulate the biology of the system, even in the presence of a biochemical milieu, and how they then play together using different transcription factors for regulation. So figured out how shear stress can play a part and how still exercise and unidirectional shear stress can help, but that still pro-remodeling shear stress that might happen because of the stiffness of the red cells, increased adhesion, and disturbing flow throughout the vascular tree might still be able to exacerbate remodeling. So how do we move that to an in vivo system? So there is Tim Towns, who was at University of Southern Alabama, no, Alabama, Birmingham, developed the Towns transgenic mouse model in the late 90s, where they knocked out the mouse hemoglobins, knocked in the human hemoglobins. There have been a few other models, but this one actually was great to recapitulate the human condition, and you can breed it with Mendelian genetics. And so we used this model, and because we were looking at vascular remodeling or arterial remodeling in children, we first went and said, well, let's sacrifice mice at 3 weeks to 1 month. So right at weaning, we sacrificed mice and isolated their carotid arteries to look at the quality of elastin content in the arteries. And again, at 3 weeks, this is just based on the genetic condition. We added no interventions here. You can see there was a degradation of the elastic lamina in the sickle cell mouse carotid arteries, again, without any other intervention. And so we quantified that, and it was quite significant. And so then we went and looked at, to ask the question if cathepsin K was being expressed in our sickle cell mouse models. And so again, AA, heterozygous, and sickle cell, we immunostain for cathepsin K, and you can see an increase in the amount of cathepsin K in the arteries of the mice with sickle cell as early as 1 month. So again, I had a collaborator on my students committee. She was a sickle cell clinician, and she had seen the J&K inhibitor data with cells, but we were like, we don't think we could put this in the animals because they'll never make it to humans. She said, inject it into the mice. We need something to help these people. Go on ahead for it. Okay. So we did systemic injection of the J&K inhibitor, SP600125, over a 2-month period. My students did daily injections over 2 months, starting from 1 month to 3 months. And to ask the question, does systemic inhibition of J&K protect the artery wall? And again, sometimes you get beautiful things, which is super exciting. So again, with vehicle-treated mice, you see from 1 month to 3 months, this is the 3-month mice. You can see the perimeter of the artery wall. You see how nice and rippled the elastic lamina is, and same thing for the hetero. But the SS mice were quite expanded, right? So we get this outward remodeling. There's a bit of duplication of the elastic lamina here, but in the mice that were treated with the J&K inhibitor, block that. And you see these structures look very similar to the wild type animal. And then again, on the closer look, we quantified elastin fragmentation. This is a Van Giesen stain, and then we did this by eye. Again, a significant difference in the SS animals that you could block with 2 months of J&K treatment. And looking at cathepsin K staining again, at 3 months, you see it up in the SS mice with vehicle, but in the animals that were treated with J&K, you see a significant reduction of cathepsin K expression where you see the protected elastic lamina. So this is actually quite exciting that there's and the reason I show this, again, I told you earlier, the cathepsin inhibitors have not made it to through the clinic. So that's why we were looking at other targets in that pathway that we could inhibit the cysteine cathepsin to still show efficacy. And again, as an engineer, we also still looked at the biomechanics of these arteries, because if you break down the elastic lamina, you'd imagine they lose elasticity and be stiffer. But that actually was not what we saw. We saw that, and this experiment, we tie the carotid artery onto this actuator here. And if you actually increase the pressure going through the lumen of the artery, then you look at the diameter of the vessel as it changes. And again, the greater the diameter change with pressure means the greater compliance, but the more resistance it is to change, then the stiffer the artery is. And this is my student Victor, who's done this work, collaboration with Rudy Gleason. And our hypothesis was that the SS mice would actually be stiffer because we were degrading the elastin. But it turned out they actually had increased compliance with sickle cell than in the wild type. And we had, I'm not wild type, heterozygous controls. We did this to have litter-mate controls for the animals. So this surprised us. This is at 3 months though. But then when we took the mice out to 5 months and continued to watch the remodeling, things really actually started to get interesting. In the vehicle-treated mice, so these mice we treated with our J&K inhibitor from 3 months to 5 months to see if we started treatment later, how would that protect the vessel at later times? Because again, you think about the clinical translation, some children don't start treatment for sickle cell until much later in life. So we wanted to know if protection of the vasculature was necessary as early as possible, or if there could still be some benefit later in life. But we didn't expect to see these vessels to look like this at 5 months. I mean, in the vehicle treated, this vessel takes on a whole new shape. It has fins, right? So this intimomedial has now thinned in the SS animals, and you can see the diameter, or I'm sorry, the perimeter of the artery is much larger. But those mice that we treated with sickle cell with the J&K inhibitor, starting at 3 months, we were able to prevent that remodeling, or mitigate, I won't say prevent, mitigate that outward remodeling that we were seeing in these animals. But then, because we were seeing this incredible thinning, we then looked at collagen 1 staining in these arteries, which we didn't look at in the 3 months. And what we found was that by 5 months, you can see we did Mason trichrome staining, and the blue indicates collagen 1 in the vessels. And you can see these SS animals by this 5 months, you see this great loss of collagen in the untreated. But with those treated with the J&K inhibitor, we're able to preserve it. We also homogenized the vessels to do molecular assay to determine collagen content. And again, you can see that the collagen is lost in these 5-week arteries. This helps to explain that increase in compliance that we were seeing. Collagen adds the tensile strength to the arteries, and as we're also losing collagen, then these arteries are now able to be blown out almost in an aneurysmal way. And so we do see, again, this increased compliance in the SS animals, but the J&K inhibition was not able to protect it when we treated from 3 to 5 months versus what we saw in the earlier stages. And so we published this earlier this year at ATVB finally, after years. And it's hard getting a sickle cell mouse colony together because these mice do not like to survive. But again, the summary there is that cathepsin expression and activity was elevated due to sickle cell disease. And it does alter the mechanical properties because these cathepsins degrade the collagen and the elastin. And the J&K inhibition can protect that from the paralytic remodeling. But there does seem to be some age-dependent impacts that we're further investigating. But again, it also links back to the clinical condition. At early ages, at those ages, like I said, 2 through 5, when they're at the greatest risk of strokes, children are at risk for more ischemic strokes, right? Due to blockages or closes off of vessels. But at older ages, like I say older, early 30s, people with sickle cell are at risk of hemorrhagic strokes. And so as we are looking at this progression in our mouse models and this loss of the collagen 1, you can see this now maybe without the mechanical strength may be more at risk for that. So we're starting to kind of track that with the human condition. But then I just wanted to touch quickly on the hemodynamics, and then I'll open up for questions. As mentioned, disturbed flow and branch points are where we saw this disturbed flow, but look at the cerebral architecture. This internal carotid artery comes in here with twists and turns, and then as it separates, things go all over the place. So we wanted to look at what are the fluid patterns in actually children with sickle cell and see if we were able to look at disturbed flow and monitor it in these individuals. And so my student, Christian Rivera, we worked with Russell Ware he sent us magnetic resonance angiograms of children with sickle cell. And we did computational fluid dynamics where we could recreate the vessels in silico in a computer and then segment out the internal carotid artery, middle cerebral artery, and anterior cerebral artery, where we know the transcranial Doppler targets for increased velocities. And then we could apply fluid profiles and investigate what do the fluids look like there. And so one of the first things we did is a non-sickle patient and a patient with sickle. And I remember when Chris first presented these, I was like, these look really bumpy over here. Like, I think you maybe messed up. What's your smoothing algorithm? He was like, I did it all the way it's supposed to. And this was my collaborator, Alessandro Veneziani at Emory, who's a computer and math science fluid guru. So he was like, hey, man, he's doing everything correctly. And this comes back in a moment because when we apply the same fluid inputs at the internal carotid artery to all of these vessels, we got elevated velocities above 300 or 200 centimeters per second only in our sickle individuals. This one did show something over here. We can talk about that in a little bit. But we started to see that quite consistently. And we also saw disturbed flow and flow recirculation in the sickle cell arteries when we went back and looked. And that's why we started to notice that now these actually, we call these bumps or these micro features might be playing different roles. And actually they could be sickle cell adhesions, could be local pockets of neo-intimal thickening. These are things these were human samples, so clearly we couldn't go in and look, but things we want to investigate now in our mouse models. But we also wanted to look in the cerebral arteries of mice, and we saw a great publication that had a label-free magnetic resonance angiography method So we want to be able to follow the same mouse in longitudinal ways because there's quite a lot of variability among the mice with sickle cell. But this way with label free, we can follow that same mouse and watch the actual morphological changes in the carotid arteries first. And you can see here between 1 and 3 months, we saw that in the AS, the heterozygote, you see the arteries get larger, but in the SS, you can see this wide variability in how large they get, but we're able to follow them now in, I say, subject-specific ways. And we're gonna follow them out, of course, to the 5 months as well, since we started to see those greater changes. And another thing that Christian was able to do with the collaborator at Peking University in Beijing was we could use micro CT where we plastinate the vessels with the radio opaque dye. So you get all the vessels, you clear out the skull, and then in micro CT you image it, but then you can segment out your particular vessels. And this is how we're able to image the cerebral arteries themselves, which are much smaller and more challenging. And again, taking these out, we can identify the different tributaries, and then again, apply our computational fluid dynamics into these as well to match what we might be seeing. And again, ACA and the MCA, we're seeing elevated velocities. And why is this beneficial? Because then also, if we put these on our drug treatment groups, will we be able to see the changes in the vessel size or the return of the normalized velocities that we might be looking for? So close it all up, the altered hemodynamics and elevated blood velocities have been recapitulated in mice in our early studies. We are seeing age-related changes to the ECM and the effects on hemodynamics. And those, well, we published the age-related changes recently, but these now the hemodynamics will be investigated in our mouse. And again, the hope is we can match the histology, which is the biochemistry, with the remodeling and the CFD, which is the biomechanics, to really get some of the mechanisms that are the cell-mediated matrix remodeling and identify other targets that will hopefully translate into patients. So if you're studying anything else, there is a problem in sickle cell disease that you could jump on board for. And there's lots of work to be done and lots of aspects that your part of the world could be influential. So thank you for listening. Love to thank my funding sources and my lab. This is at my 10-year anniversary party they threw for me about 1.5 years ago, which was quite exciting and a surprise. So thank these people for doing the work, and I'm happy to take questions from you all. Thank you.
Wow, that was really an amazing talk. And those types of photos now will creep us all out, right? Because all of a sudden, like we're sitting so close. Very stressful. It's very stressful. But so maybe I'd like to start because I have so many questions. There were so many cool findings, and I loved the intersection between, you know, the biomechanics and the biochemistry, because you're absolutely right. So many of our studies are done. I mean, it sounds like with just no movement, no cells are moving, no blood is moving. I mean, you know, I say that myself. I know lots of investigators obviously are studying, you know, hemodynamics, but my initial thought when you were showing the data from the sort of the J&K pathways and that activation of endothelium, the red blood cells were nowhere to be found in those experiments, right? You were taking PBMCs from sickle cell individuals. So then I start to think, okay, do you think that there's, you know, does that mean the PBMCs themselves are, for lack of a better word, sort of programmed? And is that because the PBMCs are interacting with, you know, the sickle cell red blood cells, or is it that there's some other sort of almost like a circulating factor, like a metabolite, or are the sickles, because you sort of initially thought about sort of the red blood cells interacting with the endothelium. So what are you thinking in terms of, I know it's like a big question that probably doesn't have an answer.
That's a great question. And it's like, oh, I should have had a slide in there about this. Right, no, you're actually absolutely correct. The red blood cells do interact with the monocytes. Red blood cells monocyte tethering happens. Because also remember, these red blood cells, because of that damaged membrane, they are way more adhesive also. So there's increased number of cell contacts, plus thrombospondin plays a part in bridging monocytes with red blood cells. Okay. Then the hemolysis of the red blood cells activates monocytes because they're producing ROS. The monocytes are producing TNF, so they're circulating in this TNF and whatever other cytokine bath is in the blood milieu. So all of those things are happening as you are suggesting. Okay. I say we started to focus on those because when we did red blood cell studies, like isolated just red cells and study them, there, I think, are ways that the red blood cells mechanically can damage the endothelial cells, and they might also, but they don't really bind like we were seeing with the white blood cells. Because you think a PBMC, particularly monocyte, could bind and then penetrate into the wall of the vessel if it wasn't on the tissue culture, you know, plate, and then really be activated and do more inside of the artery wall. But a red blood cell wouldn't enter the wall.
Yeah, like right. So I'm wondering too, I just recently learned, and this, you know, is something, you know, I know nothing about, so I could very well be speaking, you know, completely incorrectly, but that sort of oxygen saturation of the red blood cell itself can impact sort of vasodilatory function of endothelial cells. So then I'm sort of thinking again with respect to the, not so much your PBMC endothelial models, but then the impact on, you know, how a lack of or a reduced oxygen sort of tension, so to speak, would then influence the endothelial cells. I don't know if that's something that's, you know, relevant or not, but.
Well, I think that's something to think about in vivo for sure, right? Because
Yeah.
in vivo, there is the, what is it, the cell skimming layer that keeps the red blood cells away from the ECs, but then other things can interact and there's definitely oxygen transport there. But I think when really, so I say yes, but also the reminder is that the red blood cells, even though they are able to sickle and not, they do bind oxygen, and
Yeah,
you know,
right.
there's a lower number, but they still will bind oxygen in the way that a wild type would.
Right.
But the anemia still comes into play, as you mentioned. So that would be an interesting thing for an in vivo situation to investigate.
Oh, okay.
All the moving parts.
I've taken up that. Can anyone want to raise hand and ask Manu some questions?
I have a quick question.
Lale, go ahead.
So this JNK inhibitor is really nice, you know, and hopefully one day it will become available in humans. My question is really simple. Does that JNK inhibitor, does it have an effect on red blood cell morphology, or, you know, that sickle cells association with the vessel wall, or is the effect is purely on the vessel wall? So
yeah, that's so I breezed through that kinase bioplex thing we did. We did combinations to look at if we had just the endothelial cells alone, we treated with JNK, or I'm sorry, the signals of ECs alone, ECs plus TNF, monocytes with endothelial cells, no TNF, monocytes with endothelial cells plus TNF, right? And so we saw that the monocyte binding to the endothelial cells actually helped turn on JNK signaling at a lower level. And then TNF also turned it on at a low level. So then together they turned it on. So we think that the monocytes are inducing the endothelial cells to make the cathepsins because of that binding. And then we did a separate study where we blocked the TNF receptor on the endothelial cells, and we did not get the increased amount of cysteine cathepsins in the cell lysate. Because again, that lysate will have monocytes and endothelial cells when we lyse them, right, of those that have adhered. So we think that the monocytes are turning on the endothelial cells to produce it. Now, alternatively, though, what I think could also be happening in vivo is when monocytes transcytose and enter the vessel wall and differentiate into macrophages produce tons of cysteine cathepsins.
Right.
But we think for the timeline of the studies we have here, it's turning on the endothelial cells because there wasn't really monocyte differentiation. That's the working hypothesis we have with that right now, though.
Yeah, because those images you showed of the vessel wall with that sort of just glowing cathepsin, right, was really like, presumably that's, I mean, it's not just endothelial cells, obviously at that point, it's muscle cells and presumably macrophages or something that have accumulated as well.
Well, that's been the challenging part. This kind of part that's like, again, where mouse and human physiology becomes different is we have actually tried to label FR80 in our arteries, and at least in the 1 and 3 months, we haven't been able to see it. Then the 5 months gets so thin, it's like you'd imagine you would have seen it. It was so we're wondering in our mouse carotid artery models if we are really seeing monocyte accumulation, because from the human condition, there's been some. I'm going to say it interesting. There's been great work that has hypothesized that monocytes are entering and assisting in that neointimal formation, but I haven't seen it immunohistochemically labeled specifically, right? It's like that's the concept the field is moving forward, but and who am I to doubt leaders in the field? But we have not seen the monocytosis in the artery wall of our mice, and that's been a bit of a bummer for us, actually.
Well, it's not in some ways so different than in the like atherosclerotic artery, where there's already a smooth muscle cell layer before there's a macrophage monocyte layer, which mice don't have in the same way. So there could be sort of
that's a good point.
Fundamentally missing in the mice to kind of precipitate that initial accumulation as rapidly maybe as the humans, which do have the neointima. There's a much more smooth muscle cell layer, even in just diffuse intimal thickening, right, is almost no back.
I'm going to steal that to explain it to others next time.
Yeah, sure.
Thank you for that. That's helpful. Yeah.
Great. Any other questions? Raise your hand if so, figuratively or literally. Okay. All right. I think that's it. So unless there are other questions, I would like to thank you, Manny. That was excellent, excellent, excellent. That was a really, we're definitely not, I'm not used to thinking about the biomechanics of things. And I think it's such an important area to incorporate, an added layer of complexity that, personally, I'm probably not quite ready for, but you managed
to reach out if you have a loose question. And I'll be like,
yeah, absolutely. Okay, so
thanks a lot. Thanks, everyone.
Everybody. Okay, take care. Bye.
All right, be safe. COVID is real.
Bye.